Hello friends, in this lecture we'll talk about proteins. Here is a question which was asked in one of the CSR examination. Which one of the statements on protein conformation detailed below is incorrect? And the statements are pertaining to the secondary structure of the proteins and the dihedral angles that are found in the structure of proteins. So you have to identify which one is the incorrect one. Another question is based on the Ramachandran plot, which was asked in the December 2017. The phi and psi values of a beta strand composed of all D amino acids will mainly occupy which quadrant in the Ramachandran plot. So you have to select an appropriate option. So for this, we need to have some of the information on the Ramachandran plot, secondary structures, dihedral angles, and so on. So let us have a look at some of the basics of the proteins. Proteins are the polymers of amino acids which are linked to each other by peptide bonds. They can be functional or structural in nature. Some of the proteins, they have catalytic functions, and so these are called as enzymes. Enzymes, these are proteins, they are synthesized on ribosomes to the process of translation process. So the messenger RNA carries the message for the sequence of amino acids that are needed to be synthesized on the ribosomes where they are translated. There are more than 300 amino acids found in nature in the cell, and among these only 20 are repeatedly found in the structure of proteins. So these are called as standard amino acids. If we look at the general structure of the amino acid, the amino acids all have a carboxyl group and an amino group and a side chain which is designated as R. So depending on the type of side chain that is present in the structure of amino acid, they are classified into different groups. Here is the structure of lysine that is shown, which has a side chain which contributes to the positive charge as it has an amino group which can get protonated. Like sugars, the amino acids, they also can have stereoisomers. L and D form of the amino acids can exist depending on the chiral center that is present in the structure of amino acid. Except glycine, all the amino acids have at least one chiral center, and so they can have the stereoisomers, whereas glycine, it doesn't have any stereoisomers. Nearly all the biological compounds with a chiral center occur naturally in only one stereoisomeric form, either D or L. The amino acid residues in protein molecules are exclusively L isomers, whereas D amino acid residues have been found in only a few, generally small peptides, including some peptides of bacterial cell walls and certain peptide antibiotics. The table is shown which indicates the different types of different groups of amino acids. The grouping is done on the basis of the type of functional side chains that are present in the structure of the amino acid. So likewise, the amino acids can be classified into those which have nonpolar aliphatic side chain, those which have aromatic side chain, those which have uncharged R groups, those which have positively charged side chains, those which have negatively charged side chains. So these are the different amino acids. The three-letter abbreviation is routinely used to refer the amino acids, and the single letter abbreviation is now frequently used to indicate the sequence of the amino acid for a peptide chain. Let us have a look at the structure of these amino acids. The nonpolar amino acids, the amino acids with nonpolar aliphatic side chain are shown: glycine, alanine, proline, valine, leucine, isoleucine, and methionine. These are the amino acids which have the nonpolar aliphatic side chains, whereas those which have aromatic side chains include phenylalanine, tyrosine, and tryptophan. These amino acids contribute to the absorption in the UV range, as these amino acids, they have lambda max in the UV range. The polar uncharged side chain containing amino acids include serine, threonine, cysteine, asparagine, and glutamine. So these amino acids, they have polar side chains. And the amino acids which have positively charged side chains include lysine, arginine, histidine. Lysine and arginine, they have an amino group in the side chain, whereas the imidazole ring that is present in the structure of the histidine can contribute to the positive charge when this nitrogen gets protonated. The amino acids with negatively charged side chain include aspartate and glutamate, which have an additional carboxyl group in the side chain, which contributes to the negative charge. Amino acids have several properties, and one of them is that they can act as acids and bases. When an amino acid is dissolved in water, it exists in solution as the dipolar ion or zwitter ion, which is often called as hybrid ion. A zwitter ion can act as either an acid or a base. So a zwitter ion can donate a proton and thereby it can act as an acid, whereas a zwitter ion can accept a proton, thereby acting as a base. Substances having this dual nature are called as amphoteric compounds and are often called as ampholites. A simple monocarboxylic amino acid such as alanine is a diprotic acid when fully protonated. It has two groups, the carboxyl group and the amino group, which can yield protons. So the amino acid at very low pH can have a net positive charge, and when the pH increases, the carboxyl group may get deprotonated, resulting in the amino acid which has a net zero charge. Further increase in the pH results in deprotonation of the amino group, resulting in a net negative charge to the amino acid. Amino acids, they have characteristic titration curves. So acid-base titration involves the gradual addition or removal of protons. So here the figure indicates the titration curve of a glycine amino acid, and it can be seen that there are two distinct stages in the titration curve of the amino acid glycine, which indicates the deprotonation of the carboxyl group and the deprotonation of amino group. So when the pH increases initially, at very low pH, the glycine will be protonated at the carboxyl group and at the amino group. So when the pH increases, the carboxyl group loses the proton and the amino acid gets converted into a zwitter ion. So this further increase in the pH can result in deprotonation of glycine from the amino group. So the amino group now loses the proton, and you can see the pKa value for the carboxyl group in case of glycine is 2.34, whereas the pKa value for the amino group in glycine is 9.6. The sharp point of inflation is seen when all the amino acid, when all the glycine molecules are deprotonated at the carboxyl group, and further increase in the pH results in sharp inflation, and then the amino group starts losing the proton. So the this point, midpoint of the inflation region, is referred as PI, that is called as isoelectric point, which can be calculated from this formula pk1 plus pk2 upon 2. So another information that is obtained from this titration curve is that the glycine can act as a buffer in the 2 pH range, one pH near the 2.34 and the another one near 9.6. So glycine is a good buffer in these pH regions. If we talk about the pKa value of the carboxyl group in the acetic acid, the pKa value for that carboxyl group is 4.8 in acetic acid, whereas in case of glycine, that pK value is 2.34. It is due to the environment that is present in the glycine molecule, which is contributed by the other atoms. So here the repulsion between the amino group and the departing proton lowers the pKa for the carboxyl group, and oppositely charged groups lower the pKa by stabilizing the Zwitter ion. If we look at the pKa value of the amino group in the methyl amines, the pKa is 10.6 for amino group. In case of glycine, the pKa value for the amino group is 9.6. That lower pKa for amino group is due to the electronegative oxygen atoms in the carboxyl group, which pulls the electrons away from the amino group, thereby lowering its pKa value. So the environment around the carboxyl group and the amino group contributes to the lowering of the pKa value of these functional groups in the amino acids. For glycine, which has no ionizable group in its side chain, the isoelectric point is simply the arithmetic mean of the two pKa values, pKa of carboxyl group and pKa value of the amino group. So it comes to 5.97 for glycine. So here the table indicates the different amino acids. The pK value of the carboxyl group and the amino group are shown, and the isoelectric point for each amino acid is also shown. You can see that some of the amino acids which have the ionizable side chains, these include only the 7 amino acids which have the ionizable side chains, and they also contribute to the pK value. So that was about the amino acids. Peptides are chains of the amino acids which are linked to each other by peptide bond. So two amino acid molecules can be covalently linked through a substituted amide linkage termed a peptide bond to yield a dipeptide. Such a linkage is formed by removal of the elements of water from the carboxyl group of one amino acid and amino group of another. Peptide bond formation is an example of a condensation reaction, a common class of reaction in living cells. So you can see the condensation reaction between the two amino acid occurs, resulting in loss of a water molecule and joining of the two amino acids through the peptide bond. Three amino acids can be joined by peptide bond, resulting in tripeptide. So if four amino acids are linked, it is called a tetrapeptide. Five amino acids can form pentapeptide, and so on. So those amino acids which have those peptides which have the amino acids in the range of 2 to 18, they are called as oligopeptides, whereas those peptide chains which have amino acid residues more than 20, they are called as polypeptide. In all the peptides, there will be a terminal end which has a free amino group, and another terminal end which has a free carboxyl group. So this gives orientation to the structure of the protein. So accordingly, these are referred as amino terminal end and carboxyl terminal end respectively. Peptide bonds are planar. In the late 1930s, Linus Pauling and Robert Corey embarked on a series of studies that laid the foundation for our present understanding of protein structure. They began with a careful analysis of the peptide bond. The carbons of adjacent amino acid residues are separated by three covalent bonds. So you can see the C alpha of each amino acid, adjacent amino acid, they are separated from each other by three covalent bonds. Here is the resonance that is shown, that among the CN and the oxygen of the carbonyl carbon, the electron cloud can resonate, which results in a dipole, and hence the CN bond, the peptide bond, is double bonded, partially double bonded in nature. So that is further noticed from the data that the rotation is not permitted across the CN bond, whereas rotation is permitted across C alpha C and NC alpha bonds. Here is the figure indicating the bond lengths. The peptide bond is having 1.32 angstrom bond length, and the angle of rotation is also shown. The NC alpha and rotation across NC alpha bond is referred as phi, and the rotation across C alpha C bond is referred as psi. So these phi and psi, these are the dihedral angles or torsional angles which are necessary for understanding the structure of the protein. So these phi and psi values can have any value between minus 180 degree to plus 180 degree. The 6 atoms of the peptide group, they lie in a single plane with the oxygen atom of the carbonyl group and hydrogen of the amino nitrogen, they are trans to each other. Rotation is not permitted across the peptide bond, and the rotation is permitted across NC alpha bond. Rotation is permitted across NC alpha bond and C alpha C bond. Rotation is not permitted across the peptide bond because it is partially double bonded in nature. Ramachandran plots are used for studying the structure of the amino acids, which possible structures are possible in the protein structure. So phi and psi can have any value between minus 180 degree to plus 180 degree, but many values are prohibited due to the steric hindrance between atoms in the polypeptide backbone and amino acid side chains. The permitted values of phi and psi are usually indicated in the 2D map of the phi-psi plane, also known as Ramachandran plot. You can see over here the figure which is showing the Ramachandran plot. The phi angles and the psi angles are plotted, and now these are the data are collected from the X-ray diffraction analysis of the proteins, and the phi and psi values are calculated, and you can see that in some regions the red zones are shown, which indicates that here the conformations are possible with less steric clashes or no steric hindrance is seen in these regions. Whereas the yellow regions are also shown, which indicate that slightly shorter van der Waals radii are used in the calculation, and here also some structures are possible. Whereas the regions which are shown white, they indicate that there the structures are not possible because the atoms can contribute to this steric hindrance, because of which the structure get destabilized. So here in the upper left quadrant, now this Ramachandran plot is for the L amino acid that is shown over here. The Ramachandran plot for the D amino acids will be different. So we'll talk about that. So for L amino acids, the beta sheet structure can have the phi and psi values in the upper left quadrant, whereas the right-handed alpha helices will have the phi and psi values in the lower left quadrant, whereas the left-handed alpha helix, it can have the phi and psi values in the upper right quadrant. In case of D amino acids, the opposite will be seen, that is beta sheet will be seen in the lower right quadrant. The right-handed alpha helix for D amino acids will be seen in the upper right quadrant, whereas left-handed alpha helix for D amino acid will be seen in the lower left quadrant. So this has to be kept in mind. Here is the table indicating the phi and psi values for each secondary structure. So antiparallel beta sheets will have the phi values minus 139 degree and psi value plus 135 degree. Parallel beta sheets will have minus 119 degree and psi value as plus 113 degree. Right-handed alpha helix has minus 57 phi value and minus 47 degree psi value. Likewise, all the others are shown. Left-handed alpha helix has a phi value of plus 57 degree and psi value of plus 47 degree. So this was about the Ramachandran plots. Proteins, they can have the secondary structure. Primary structure of the protein is the amino acid sequence of the protein, whereas secondary structures are they occur due to the internal bonding between the amino acid residues which are situated close to each other. So these secondary structures include alpha helix, beta sheet. Alpha helices can be either left-handed or right-handed. Alpha helix. It is a rigid rod-like structure that forms when a polypeptide chain twists into a helical conformation. Alpha helix can be right-handed or left-handed. Right-handed helices are energetically more favorable. In almost all the proteins, the right-handed alpha helices they predominate. So the length of the alpha helix is about 10 to 15 amino acid residues, and the per helical turn there can be 3.6 amino acid residues. The pitch, which is called as the distance between the corresponding points per turn, is about 0.54 nanometer or 5.4 angstrom. Each residue is related to next one by a rise of 1.5 angstrom along helix axis. A single turn of alpha helix involves 13 atoms from oxygen to hydrogen of the hydrogen bond. Hence, alpha helix is often referred as 3.6 13 helix. It can also be that the alpha helix can have 10 atoms, and accordingly it is referred as 310 helix that we seen in the previous table of the Ramachandran plot. Intrachain hydrogen bonds form between the amino group of each amino acid and carbonyl group of the amino acid 4 residues away. Carbonyl and amino groups of amino acids at the ends of alpha helix are not hydrogen bonded. Side chains of the amino acid residues they orient outwards. So these side chains they will orient outwards, and all the hydrogen bonds they lie parallel to the axis. So this is the axis, and almost all the hydrogen bonds they will lie parallel to the axis of the alpha helix. So alpha helix can form from either D or L amino acids, but a single alpha helix cannot have a mixture of D and L amino acids. So it cannot form from a mixed copolymer of D and L amino acids. Stability of alpha helices. Amino acid residues such as alanine, glutamine, glutamate, leucine, methionine, arginine, they show higher tendency to form alpha helices. Proline or glycine, they disrupt the alpha helices. Proline lacks an amino group, and because its ring structure restricts its pi value to near 60 degree, minus 60 degree, so it gives kinks to the helix, thereby destabilizing it. Glycine forms coiled structures instead of helix because it is more flexible. Five different kinds of constraints they affect the stability of an alpha helix. One is electrostatic repulsion or attraction, which occurs between successive amino acid residues with charged side chains. The bulkiness of the adjacent R groups such as aspargine, serine, threonine, and cysteine. The interactions between the side chains which are spaced 3 or 4 residues apart can also contribute. The occurrence of proline or glycine residues in the structure can destabilize it. The interaction between the amino acid residues at the ends of the helical segments and the electric dipole inherent to the helix also can cause destabilization of the helix. The tendency of a given segment of a polypeptide chain to fold up as an alpha helix therefore depends on the identity and sequence of amino acid residues within the segment. Beta conformation organizes polypeptide chains into sheets. So Pauling and Corey predicted a second type of repetitive structure which is called as beta conformation, or often referred as beta pleated sheets, because it appears as a zigzag in nature. So beta strands can be either parallel or anti-parallel. So here one region of the polypeptide chain gets bonded with the another region or another strand of the polypeptide chain through the hydrogen bonds. So if the beta strands they run anti-parallel, the beta sheet is referred as, if they run opposite, they are referred as anti-parallel beta sheets. And if both these strands they run in the same orientation, in the same direction, it is referred as parallel beta sheets. So beta pleated sheets they form when two or more polypeptide chain segments they line up side by side. Each individual segment is referred to as beta strand, which is fully extended instead of binding. Distance between adjacent amino acid along a beta strand is approximately 3.5 angstrom in contrast to 1.5 angstrom along alpha helix. Here is the parallel beta sheet that is shown. So these beta sheets are stabilized by the interchain hydrogen bonds that form between the polypeptide backbone, amino and carbonyl groups of the adjacent strings. So you can just have a look that the hydrogen bonds in between the beta strands, amino and carbonyl group, they are slightly different from the hydrogen bonds between the beta strands in the antiparallel strands. The another kind of structure is the beta turn, which can occur due to the presence of proline or glycine. So wherever the glycine or proline is present, a sharp turn is seen, which is called as beta turn. So type 1 turn, beta turn includes the proline at the second position, whereas the type 2 beta turn involves glycine at the third position. And the first and fourth amino acid, they are involved in the hydrogen bond formation. Supersecondary structures are often seen. These are the structures which involve alpha helix and beta sheets. So depending on the supersecondary structures, different types of protein domains are found. These include, some of them are listed: beta alpha beta motif, then beta Mendel motif, in which the two antiparallel beta strands are connected by polar amino acids and glycine to effect an abrupt change in the direction of the amino acid, in direction of the polypeptide chain. In alpha-alpha motif, two successive alpha helices, they are separated by a loop or non-helical segment become enmeshed because of the compatible side chains. Tertiary and quaternary structures. So here the long-term interaction between the amino acids, they result in further folding of the polypeptide chain, resulting in the tertiary structure, and which can be further stabilized by the bonds such as the disulfide linkage and so on. So further folding can occur, and the amino acids like the proline, threonine, serine, and glycine, they contribute more to such kind of interactions. And different kinds of weak bonds such as the hydrogen bonds, then ionic interactions, disulfide cross links, which is a covalent bond, and they can also contribute to the stability of the tertiary structure. If the two polypeptide chains are connected to each other through non-covalent or covalent bond, then it gives rise to the quaternary structure. So this was about the structure of proteins. Let us come to the discussion on the questions. Which one of the statement on protein conformation detailed below is incorrect? L amino acids can occur in type 1 beta turns where phi and psi both are positive. A peptide rich in proline is unlikely to adopt a alpha helical structure. Proline residues have high propensity to occur in beta turns, yes. And the dihedral angles alpha and psi of amino acids in unfolded proteins are exclusively positive. So here the incorrect one is the first one, because here the type 1 beta turns will not have both positive. The L left-handed helix will have both alpha and psi as positive. The second one, here we saw that in the D amino acids where the beta strand can be found. So the beta strands can have the phi and C value in the lower right quadrant. So lower right is the correct quadrant for the phi and psi values in the beta strands containing of the consisting of D amino acids. So lower right is the correct option. So this was about the proteins. In next lecture, we'll talk about the other aspects of the protein. Thank you and happy learning.